Chin.
I'm Caden Chin.
Okay, and I'm Kevin Clausen, and this is our microbiome initiative relating to the gut microbiome.
Okay, so our project involved the microbiome within the digestive tract of humans. It's a very interesting part of the body because we don't know a lot about the microbiome within our digestive tracts, and there's thousands of different bacteria living within the digestive tract as well. So we know that there's different bacteria within it that can perhaps cure diseases that we get, or they can also cause the diseases that we get because they're harmful. So we chose to look at colitis ulcerosa, also commonly known as ulcerative colitis. It's a disease that is caused by unknown reasons currently, and it occurs in the digestive tract. About over 200,000 cases occur within the United States a year. So we proposed to use confocal microscopy to assist our observation of the bacterial structure and function to perhaps find the cause of ulcerative colitis, and we think that it will help greatly.
So the main question we're trying to answer is how can we apply microscopy? Is that right? Okay. Techniques to further understand how we can determine the specific microbes in the varied gut microbiome that causes colitis ulcerosa. So like Caden said, despite many tests to determine what the specific cause of colitis ulcerosa is, unfortunately, they haven't found much about what the cause is. So based on the confocal microscopy techniques that we will explain later, this is how we propose that we're going to address the issue of how to find the cause of this disease.
Okay, so first a little bit of background about ulcerative colitis. So it is an inflammatory bowel disease type. So it affects the inner lining of the colon and the rectum, so it's mostly the large intestine. And it is characterized by the ulcers in the lining, as well as diarrhea, bloody stool, and cramps.
Confocal microscopy, what mainly differs it from other kinds of microscopy is that there's a pinnacle at the end which allows out of focus light to be eliminated, therefore allowing the finest detailed structures of the specimen to be observed. And while the cause has not been seen yet of this disease, current technology advances have allowed our abilities to understand these diseases in general to achieve advancement in learning about the microbiome in the digestive tract specifically.
So research has been done before, experiments have been done on trying to find the causes of different inflammatory bowel diseases. So they've done microbiota fecal transplants. One study was done on rats where they took the fecal matter of a healthy rat and they stuck it into an unhealthy rat, and the unhealthy rat actually recovered from its illness after receiving the fecal matter from the healthy rat. So they also use diet manipulation to try and make sure that whatever they want going into their subjects is controlled. They also use probiotics and antibiotics to try and counter different bacteria within the digestive system.
By one study done by the American Society for Microbiology, they tried to observe the different microbes within the mucosal surfaces within the gastrointestinal tract. And by doing this, they were able to use confocal laser microscopy. This is going to be microscopy to allow a determination of the cells within, because with confocal microscopy, they use the staining called the fish staining, which stands for fluorescence institute hybridization. And with this, they insert fluorophore labels that allow the colors of different cells to be recognized. So therefore, groups of cells and different organelles within can be recognized based on the colors that were basically given to them and allow them to be stained and recognized. And with this, they can allow the localization of different bacteria and cells to be recognized within. So based on this study, they were able to see that within the colon there were not a lot of bacterial differences that differed within the count. So based on that, what we propose to do is that we are going to observe the structural and functional observations within colitis ulcerosa, and the way we propose to do this is by, in the digestive tract, they contain the T cells and then the possible bacteria that cause this disease. And so what we plan to do is label the T cells different colors and then stain the bacteria differently to see which ones were eliminated first. The ones that were eliminated, therefore, because this disease tries its best to eliminate the most harmful bacteria first, but overdoes it. The ones that it does first are the ones that have the potential to be the ones that influence this disease the most, and therefore the ones that were not killed off, therefore have the potential to not be likely causes of the disease. And by doing this, the ones that were killed off, this allows us to narrow down our scope because there's like 1,000 species of bacteria within the gut to allow us to determine what possible bacteria may lead to the potential cause of colitis ulcerosa.
So there's a few challenges that are going to occur within this experiment if we were to do it. People have very different microbiomes because everyone's different. They grow up in different environments, eat different foods, so they have very different bacterias, and perhaps one bacteria could be harmful in one person, whereas in another person it could be helpful to them. So another problem is that there are thousands of bacteria species living within the microbiomes of people's guts, and to find that specific bacteria that may be causing a certain disease would be very difficult because it's going to take going through thousands of bacteria. Another problem or challenge is that experimentation on animals is a little bit harder because you would need to use humans instead, because humans and animals have a different ratio of different bacterias in their digestive systems. They can have the same bacterias, but they'll have different amounts.
So there's that.
The contribution.
So, other than finding a cause for ulcerative colitis, our project would bring up certain other questions. One of them would be because, like Caden said, there are thousands of bacteria within the gut, and hundreds of them we don't even know exist. So, in our study, we will find bacteria that we've never discovered before, and they may have the potential for causing other inflammatory bowel diseases such as Crohn's disease or other colitises. Also, a second question that would come up would be, since many bacteria within the gut are beneficial, such as lactobacillus, it breaks down sugars. We would be able to figure out how this bacteria would be beneficial to the gut, and also, since we know the structure of this bacteria, we would be able to find a cure for it. And building off of that, the gut microbiome is a complex biodiverse ecosystem with each individual organism having a specific contribution to the microbiome. So we would be able to figure out how this specific bacteria would contribute to the microbiome normally, and also why it causes this disease. And these are references, so now we're open for questions.
Alex? So basically, this disease, colitis ulcerosis, compared to a lot of other diseases, many people have narrowed down the microbes enough in order to determine which ones may be the potential for causing this. But for this disease specifically, what interests us most was that this was more like unknown, and scientists have done many tests to determine the nature of the microbes within this disease, but unfortunately, there hasn't been much success into discovering that yet.
mentioned that a transplant of microbes can actually result in a curative effect. Do you see any implications of things like antibiotic treatments in possibly causing things like ulcerative colitis?
Can you repeat that question real fast?
Yeah, so you guys talked about an example where bacteria were transferred from a healthy mouse to a diseased mouse, and that actually led to a recovery of that mouse.
Yes.
So then, for example, when we take antibiotics and we wipe out everything indiscriminately, do you see a role in that potentially contributing to some of these diseases? And did you find anything discussing that in your literature?
Yeah, I actually did. So there was a study shown where what happened was they did use antibiotics on a subject, or I can't remember what animal they were using, but it actually died due to the antibiotics that they used actually wiping out everything in the microbiome that was in their digestive tract. And because they didn't have the healthy bacteria there existing to probably counter the harmful bacteria continued to multiply and just take over the digestive tract and kill it.
If there are over 1,000 different type of bacteria, how you propose or narrow down that this particular bacteria is the cause for ulcerative.
So like the stain that we discussed, the fish stain, basically we'll have to if we ever if we ever tried out this experiment, we'll have to see like how many like different species within our first like sample we see, and then we'll have to stain them each differently in order to determine how each of them are set apart, and then that's how we can determine from the T cells which ones are over time, which colors are noticeably gone because of those bacteria being eliminated first.
Do you remember anything from your tour on the first day up on the 6th floor in the MSSR lab with the robots?
Kind of.
Right, the high throughput screening facility, right? So this is an area where addressing thousands, tens of thousands, hundreds of thousands of samples, right? Where we're integrating optical detection, fluorescence detection with high throughput methods really allows you to address some of these bottlenecks, right? It's one of the reasons that we brought you up there, even though it's not a microscopy facility per se, it really overcomes some of these limits, right? Because the number of samples that you need to do these types of studies in certain cases is very high, right? And becomes cost prohibitive, right? So increasing throughput is a potential option to address what he's talking about, which I don't know if you guys remember anything from that tour, but maybe you could comment on it if you do.
Oh, there was another comment. So another way that we might be able to narrow down the scope is ulcerative colitis is an autoimmune disorder, so it does start to attack itself. And this is because the bacteria that potentially causes it, the immune system will attack it, but it doesn't stop attacking it. So in our studies, when we add the T cells to the bacteria, the T cells will just continuously attack the bacteria, while other bacteria, it'll just stop.
Have fecal transplants been used successfully in humans?
I didn't find any studies about this because I don't think anyone wants that to happen.
I thought I heard that it has been done.
It has been done.
I didn't find anything on that, so.
Here's a cure.
Yeah. Well, there's a difference between what's been done and what's been published in the literature. There are certain clinical trials and methods that are in process in certain areas that haven't yet been published per se. So I think most of the students are leveraging either the web to look at the scientific literature, but that stuff is ongoing and has some pretty strong positive implications. It does speak though to the realities of people getting over what it actually entails.
Well, and eventually, if you could really identify things, you can always improve the treatment and provide just the bacteria, for example, right? But you have to learn that first. So you start with the materials you have to work with, which right now, since we don't have these techniques fully in place, you can't isolate exactly which bacteria were the curative ones. So you work with some kind of bulk, person-to-person transplant using this material. Later, if you really refined and understood it, you can get rid of all of those details and just provide the curative aspect of what it was. You don't need to do that blunt force version of it.
Another question? I was just more of a comment
Oh, so I was.
There are, as some people have been discussing, there are really strong links between the gut immune system and the overall immune system. You think about your gut processes all sorts of foreign material that the rest of your body would freak out in exposure to it. And so how your immune system in the gut translates to your overall immune system is not fully well understood, but it's known there's very strong linkages. So it could be that they're basically activating some immune process in the gut that then actually then goes out and activates other immune processes or gives you antibodies that are developed in the gut that then can proliferate through the rest of your body and give you a cure for a virus, for example. So that's why it's very, very interesting to study gut immunology versus total immunology, as one of our first groups mentioned.
I got one question. This isn't really specific to this, but there's a lot of overlapping non stuff. I was wondering, is there like a cloud for this that people share, or is it so guarded that, like you mentioned about it's not being published or what and all that stuff? Is it so guarded that they don't put it up in the clouds for other people to? You know what I'm saying?
Yeah, I know what you're saying. I mean, it raises a broader ongoing discussion in the scientific community about open access to the data that you all fund through your taxes. Right? And large publishing companies make a lot of money on keeping it behind paywalls. Right? So the National Institute of Health, the NSF, major funding agencies are requesting and requiring that we make data available through open access repositories. The UC system actually requests that all of our pre-print publications are uploaded to a server that's available to anyone to use, but it's really something that's only been in the discussion in the last, meaningfully in the last 5 to 10 years or so, because there's so much scientific literature out there, and with scientific literacy of the general populace and interest increasing, and the sort of methods we have for sharing data online, there's a lot more demand for people to really have access to this stuff, but it's just not the traditional approach. It's usually behind, you can only get in if you're on campus of a university or if you have a subscription to a journal. That's slowly changing. There's a lot more open access publications, but the model is one that's under a pretty intense discussion in science in general about how to make the data available.
Yeah, because of the acceleration of AI and all that stuff, that could greatly help and all that stuff. There was like a cloud and all that stuff.
A lot of people are being much more open to sharing their data more broadly to leverage these sorts of, whether you talk about cloud-based crowdsourcing, scientific crowdsourcing, right? There's other approaches to things, but it's really an emerging area of people figuring out how to strike the balance between, making sure that the science is well vetted and peer-reviewed, but making it accessible not only to the general community, but also to other scientists in a way that doesn't jeopardize the individual research integrity, right? So that it's a very vigorous dialogue right now, but it's something that's still not where you might think or hope it will be.
Another question for presenters? Yes?
This is kind of on that vein. You keep talking about how spinal taps are so nuanced and whatever else. Is there something sort of like, when you get your license, you're a body part donor or an organ donor, where when you're having one of these tests that's so vile that you would sign something so that the rest of the materials in that test could be utilized by other scientific communities, or is there anything like that?
There actually is a building movement to have people, and many people can donate their bodies for science. Often that movement
I'm talking
is
about like
studied.
an active person who is having a spinal tap. Part of that fluid could be used for some other.
Yeah, so there's one where you're talking about like organ donation and and actually donating the whole body, right? And on the other side, yes, when you do procedures, you can sign off to allow scientific researchers to have access to those materials as well. So yeah, collecting samples from hospital settings and from medical practice is actually very ongoing. So one of the things that powers a lot of research here at UCLA is we have a medical system. And so we can go get biopsy samples and then take out some tissue from a brain. Like they mentioned looking at motor neurons and doing biopsies there, right? When you do, for example, a resection during cancer, you will get some healthy tissue as well. And so dividing up those samples to researchers. Now the patients have to make sure they acknowledge and authorize that, right? And then of course their identities have to be protected, and there's all sorts of protocols on top of that. But that's definitely a well-established practice, and also something that's increasingly being spread to different types of tissues. And there's also tissue banks where people can send wholesale tissues, because some problems can only be addressed when you collect 10,000 tissue samples from diverse groups of patients with and without the disease.
So is it standard procedure that if you're having one of these tests that you're given a proposal to sign off on it?
It's
Or is it
growing.
not?
It's growing.
Where you are.
Yeah, and whether you're a research hospital or a regular hospital makes a huge difference oftentimes too.
All right. Well, thank you guys
Thank
very
you.
much.